Welcome, curious minds, to another deep dive. Today, we're plunging into a kingdom that's, well, far more diverse and impactful than many of us realize: the Eumycota, or true fungi. Forget everything you thought you knew about just mushrooms. Our mission today is to equip you with a concise, engaging understanding of these incredible organisms, straight from the insights of a foundational text, the Fifth Kingdom. We'll be walking through their unique characteristics, their amazing adaptations, and focusing on three key phyla: Zygomycota, Glomeromycota, and Microsporidia. Think of this as your essential shortcut to really getting a grip on the fungal world.
Indeed. It's often said that for every known fungal species, there are maybe 6 more just waiting to be discovered. We're talking potentially well over 1 million species out there. So while we often visualize a mushroom, that's just a tiny fraction, really, of what true fungi are. Our goal today is to bring this unseen world to life, explaining the how and why behind their, ecological dominance.
Okay, let's unpack this then. What makes fungi such evolutionary winners? I mean, they're everywhere, doing vital things, from breaking down dead stuff to fermenting our food and drink. What's their core secret?
Well, if we look at the big picture, one of the most fascinating things is their radical evolutionary path. Most true fungi lack motile cells, cells that can move on their own. That tells us they've evolved to thrive on land for a very, very long time. And this commitment to a terrestrial lifestyle, it opened up far more ecological niches, more substrates than water ever could. It was a real game changer for them.
And they're heterotrophic, right? Meaning they can't make their own food like plants do. That sounds like it could be a disadvantage, but clearly it hasn't held them back.
Not at all. And that raises the question, doesn't it? How do they thrive if they can't photosynthesize? The answer really lies in their specialized enzymes. They've evolved this incredible biochemical toolkit. They can digest extremely tough organic materials that are, well, largely unavailable to other organisms. We're talking things like chitin.
Like insect shells.
Exactly. And keratin, skin, hair, feathers. Cellulose, which is most plant debris, and lignin, a major part of wood. This unique ability makes them the world's premier recyclers, absolutely essential for breaking down the huge amounts of plant waste produced every year. Without them, we'd be buried in it.
So not just a cleanup crew, they're like the ultimate biorecyclers. Okay, but what are the physical tools, the structures that make all this possible?
Their incredible success really boils down to 2 key innovations: the fungal spore and the fungal hyphae. Let's start with spores. These are non-motile microscopic reproductive units. Think of them like highly specialized seeds, maybe. They come in this dazzling array of forms, each designed for a specific function, and they're produced really quickly in enormous numbers.
So dispersal, right? Yeah. Wind, water.
Wind, water, animals, you name it, allows for incredibly efficient dispersal. But what's really remarkable is the resilience. They can survive long periods of freezing, starvation, or desiccation that's extreme drying out, spelled with 1 S, 2 Cs. Fungal spores are truly ubiquitous. You find them in astronomical numbers in the soil and thousands per cubic meter just in the air we breathe.
Wow. So they're the ultimate tiny travelers and survivors just everywhere. Now, tell us about the hyphae, the engines of fungal growth.
Right, the hyphae. These are the vegetative or growing structures of most fungi. When a spore germinates, it sprouts 1 or more of these hyphae, and they grow continuously, but only at their tips, exploring the microscopic world around them. Picture a microscopic branching root system, maybe, but instead of just anchoring, it's actively seeking out and digesting nutrients. These hyphae branch repeatedly, forming this network we call a mycelium, to explore their surroundings really efficiently.
And these tips are pretty special.
Oh, yeah. Each hyphal tip is strong, it's waterproof, and it's made of chitin, that same tough polymer found in insect exoskeletons. And right at these growing tips, the fungus secretes digestive enzymes directly into its environment. Plus, they can exert hydrostatic pressure.
Pressure, like pushing their way in.
Exactly. It allows them to actively penetrate and exploit solid substrates. It's a feat most bacteria, their main competitors in recycling, they just can't match that physical penetration.
That's amazing. So they're not just growing outwards, they're actively interacting, like miniature biological drills, breaking down and absorbing stuff right at the tip.
Precisely. If you could zoom way in with an electron microscope on a hyphal tip, you'd see these tiny sacs, they're called vesicles, all concentrated at the very front. These vesicles are packed with new wall material and those digestive enzymes ready to go. And just behind them, you'd find the mitochondria, the cell's energy factories, fueling the whole process. This highly specialized microscopic growing engine at the tip is why fungi are such unparalleled explorers and penetrators of tough organic matter. They're literally resculpting the world at a cellular level.
It really sounds like they're built for extreme survival. They can handle pretty much anything Mother Nature throws at them.
They certainly are. Fungi have learned to cope with an astonishing range of environmental extremes. It's quite incredible. They can grow in temperatures from as low as -5 degrees Celsius up to 60 degrees C. Many are remarkably zero-tolerant, meaning they can thrive in incredibly dry conditions. Some molds can grow where most plants would just wilt and die.
Really?
Don't try. Oh, yes. And others tolerate oxygen levels as low as 0.2%. They can survive in environments ranging from extremely acidic, like pH 1, to quite alkaline, say pH 9. This adaptability is, well, it's truly astounding.
So what does this all mean for us then? This incredible adaptability, this enzymatic power. It makes them both incredibly beneficial, but also sometimes problematic, right?
Absolutely. On one hand, you have the saprobic fungi, those that live on dead organic matter. They are recyclers par excellence, absolutely vital. But they're also opportunists. Their spores are everywhere, as we said, and they'll germinate wherever there's moisture. So they'll attack food, fabric, paper, pretty much any organic matter we might want to preserve. And some even produce dangerous metabolites called mycotoxins. These can be extremely hazardous, even carcinogenic if they contaminate our food supplies.
Yeah, that's the downside.
And then you have the parasitic fungi. They cause the majority of serious plant diseases globally, and also some significant illnesses in animals and humans too.
But as you said, there's a much brighter side to how fungi interact with human affairs. We've actually harnessed their biochemistry for ages.
Indeed. Think of the classics. Beer, wine, bread, gourmet cheeses, soy sauce, all products of fungal fermentation. We owe them a lot. Beyond food, they produce vital antibiotics. Penicillin, of course, being the most famous. Also immunosuppressants, organic acids, a whole host of useful chemicals. They can even convert plant waste into high-protein animal feed or environmentally friendly packing materials. And of course, many are delicious food sources themselves. Mushrooms, chanterelles, morels, truffle.
Truffle.
Exactly. And what's fascinating now is that even some of those parasitic forms are being recruited for biological control. We're using fungi to fight insect pests, weeds, even other harmful fungi. It offers sustainable solutions to agricultural challenges.
And we absolutely cannot forget the silent, invisible powerhouses that form mycorrhizas. You mentioned these are intimate, often essential associations with the roots of almost all higher plants.
Yes, absolutely critical. Representing one of the world's oldest and most successful forms of mutualistic symbiosis, they are truly the unsung heroes of terrestrial ecosystems. A partnership that underpins so much of life on land, forming this vital bridge between the plant and the soil that's hundreds of millions of years old.
Incredible. Okay, let's zoom in now on our first specific phylum, Zygomycota. This group is famously diverse, but also, as the chapter notes, maybe the least well-defined scientifically right now. What are its key features and why is it being reevaluated?
You're right, the Zygomycota is, well, it's a bit complex. It's considered a non-monophyletic group.
Meaning not a single clean branch on the evolutionary tree.
Exactly. It doesn't quite fit into one neat lineage, which is why scientists are currently reclassifying parts of it. However, many members, particularly in the subphylum Micromycotina, are very common. They're fast-growing saprobes. They quickly colonize substrates rich in accessible carbon, like sugars or starch. Think of them as the first responders, the initial colonizers when a new food source appears.
And the name, Zygomycota, that comes from its unique sexual reproduction, doesn't it?
Yes, it does. The name is derived from the zygosporangia. These are thick-walled, very resistant structures formed during sexual reproduction. Zygos is Greek for a yoke, like for oxen or just joining. In this process, specialized cells called gametangia, these are the cells that fuse in sexual reproduction from compatible hyphae. Now, these can be from a single organism in homophilic species or two different strains in heterophilic species. They physically fuse or conjugate.
Okay, so they join up.
Right. Imagine two hyphaes reaching out, developing these finger-like outgrowths. They exchange chemical signals to check compatibility. Then their tips fuse, the walls break down, and their multinucleate contents mix together. This mixture then gets sealed off by cross walls, or septa, and the paired nuclei inside fuse to form that thick-walled zygosporangium. These often act as robust resting spores, surviving harsh conditions, and you can usually recognize them by the two empty suspensors that held them up, like that yolk. Crucially, there's no difference in size or shape between the fusing gametangia, so we just label the compatible mycelia as plus and strains, no males or females here.
Got it. So those zygosporangia are the sexual structures, but the chapter also mentions anamorphs. What are those, and how common are they compared to the sexual phase?
Ah, good point. You actually won't often see Zygosporangia just out in nature. They can be quite rare. The asexual or anamorphic phases of zygomycetes are far more common and much easier to find. Think of the fuzzy mold on bread or peaches, or the mold growing rapidly on animal dung. Those are often zygomycete anamorphs. They're efficiently producing non-motile asexual spores called mitospores inside specialized structures called mitosporangia. They're the workhorses, really, for rapid reproduction and getting around.
Let's delve into some of the more spectacular examples then. Beyond the common bread molds, Zygomycota has some real specialists with incredible adaptations, right?
Indeed, it does. Consider Pulobolus crystallinus. It's atypical, but absolutely fascinating. It's coprophilous.
Meaning dung-loving.
Exactly. Dung inhabiting. It's a member of the order Mycorrhalis. It grows incredibly fast, one of the first fungi you'll see on fresh herbivore dung. It's unbranched sporangiophores. The stalks holding the spores are maybe 2 to 4 centimeters tall, and they have this unique explosive dispersal mechanism. Oh yes, the entire sporangiophore is basically one single giant cell. Beneath the black apical mitosporangium, the spore sac, at the top, there's a lens-like swelling called the subsporangial vesicle. And at the base of this vesicle is a light-sensitive retina. This actually controls the sporangiophores' growth, aiming it precisely toward light. It's phototropic. As osmotically active compounds build up inside, the hydrostatic pressure within the sporangiophore and vesicle can reach over 100 pounds per square inch. It's immense.
Wow.
Eventually, this pressure causes the vesicle to explode violently. It hurls the black sporangium up to 2 meters, aiming straight towards the light. The sporangium then sticks to whatever it lands on, thanks to some sticky goo inside. Now, this raises the question, why would Pulobolus need such a powerful aimed cannon for its spores?
To get onto grass, right? So cows eat it.
Precisely. It's a remarkable adaptation to ensure spores are launched clear of the dung pile and onto surrounding vegetation. Grazing animals then eat the grass, spores and all, and the spores pass through the digestive tract unharmed, ready to germinate in the fresh dung. It completes the life cycle. It's just brilliant biological engineering.
That is an astonishing feat. A fungus with a high-pressure, light-guided spore cannon. Okay, what about other unique strategies in this group?
Well, speaking of intricate adaptations, some members of the Mycotina subphylum take complexity to another level entirely. Take Spirodactylon. This is a fungus that grows on mouse and rat dung. Its tall, branched sporangiophore is repeatedly thrown into these tight coils. Looks really strange.
Yeah.
Within these coils, you get even more elaborate structures. Sporocladia, which bear rows of lateral cells called pseudophylalides, and each of those produces a single-spored sporangiole, basically a tiny one-spore packet.
Okay, coils, branches, pseudofungies. Why all that complexity?
Well, this strange coiled configuration makes perfect sense for its habitat. Rats and mice are omnivores, right? It's hard for spores just landing randomly to get onto their food. So the fungus's coils act like little Velcro strips. They get entangled in the animal's fur as they run along their well-trodden paths. Then when the animal grooms itself, it ingests the spores. It's a clever, if maybe slightly gross, strategy for getting back inside the gut, as the book says.
That's incredible. A fungus actively setting traps in animal fur to get re-ingested. And then there are the harpalales and acellariales, even more specialized.
Yes, these used to be lumped together as Trichomycetes, though that's now seen as more of an ecological grouping. These are eccentric groups that live almost exclusively attached to the gut lining of living arthropods, insects, crustaceans, millipedes. You probably won't encounter them often unless you're specifically looking, but they're another testament to fungal opportunism and the dedication of mycologists who study them. Under a microscope, you'd see characteristic structures like trichospores, which have these hair-like appendages, not flagella, something different. These, along with their developing zygospores, confirm their place within Zygomycota. They're just fascinating examples of extreme adaptation to living inside another organism.
Wild. Okay, finally, within Zygomycota, we have the entomophthalales, famous for attacking insects.
Right, the entomophthalales. These include some incredible insect pathogens. Entomophthora muscae is a classic example. It infects and kills houseflies. You might have even seen this. Dying flies often crawl up to exposed spots, like window panes. Then the fungus bursts through their exoskeleton, producing these tightly packed masses of sporangiophores. Each one bears a sticky, single-celled mitosporangium that gets forcibly shot away when mature. It creates this whitish halo of spores on the surface where the fly died.
Ah, I think I have seen that.
It's quite common. Those spores can then infect other unsuspecting flies that land nearby. This makes them, and related species, highly promising candidates for biological control of insect pests, a natural, targeted alternative to chemical pesticides.
Very cool. Okay, let's move on to our next phylum, the Glomeromycota. These were once sort of tentatively placed in Zygomycota, but they've since been recognized as their own distinct group, and they are incredibly important ecologically.
They absolutely are. Connecting this to the bigger picture, the Glomeromycota are the arbuscular mycorrhizal fungi, or AM fungi, sometimes called endomycorrhizal fungi. They establish these obligate, mutualistic symbioses partnerships where both benefit with perhaps 90% of all higher plants. That makes them profoundly important for ecosystems worldwide. They are truly a cornerstone of plant life on Earth, a partnership going back hundreds of millions of years.
So obligate means they have to have the plant partner, right? They can't just live on their own in the soil.
Precisely. They won't grow an exenic culture that just means a pure culture all by themselves without any other organisms. They absolutely must be associated with a living plant root. Their resting spores, which are generally very large and thick-walled compared to other fungi, are common in most soils. And these spores are actually stimulated to germinate by chemicals released from nearby plant roots. It shows they have very wide host ranges, they're not too picky, almost any plant root will do.
Okay, so the spore germinates near a root. How does the partnership actually work inside the plant?
Well, they're hyphae, which are usually non-septate, meaning they lack regular cross walls, spread through the soil, and then they penetrate the living root cells of the plant. They actually go inside the cells. And inside these root cells, they develop these diagnostic, finely branched, tree-like structures called arbuscules.
Arbuscules like little trees.
Exactly. That's what the name means. These arbuscules are the critical interface. They massively increase the surface area between the fungus and the plant cell membrane. This is where the fungus efficiently exchanges mineral nutrients it has gathered from the soil, especially phosphorus, for photosynthase, the sugars and other organic compounds the plant makes through photosynthesis. Many Glomeromycota also produce lipid-filled storage structures called vesicles, or sometimes intramatrical spores inside the plant roots.
It's particularly good at getting phosphorus.
Incredibly good. Their extensive soil mycelium is extremely efficient at mobilizing insoluble phosphorus, which plants often struggle to get, and moving it, along with other key mineral nutrients, directly to the plant via the arbuscules. Since phosphorus is often the limiting nutrient for plant growth, especially in natural soils, AM fungi help plants thrive in poor conditions. This makes them vital in natural habitats and gives them immense potential value in sustainable agriculture, possibly reducing our reliance on synthetic fertilizers.
Amazing partnership. Okay, finally, let's turn our attention to the smallest phylum we're covering today, the Microsporidia. These sound quite different even among the fungi.
They are quite distinct, yes. The Microsporidia, there are about 1,500 known species, are all obligate intracellular parasites of animals. They primarily infect insects, but also fish, crustaceans, and yes, even humans in some cases, particularly immunocompromised individuals. They represent an extreme example of evolutionary reproduction. They've lost many typical eukaryotic features, notably mitochondria. They don't have their own powerhouses, and they lack flagella. They reproduce mostly by forming these extremely resistant spores, which are key to their survival outside the host and for transmission.
So, tiny reduced parasites, how do they actually manage to infect their hosts? What's their strategy?
They employ a really remarkable, almost sci-fi infection mechanism. When an animal ingests a microsporidian spore, or it comes into contact with a host cell, the spore rapidly everts or extrudes this long, hollow tube called a polar filament, or polar tube. Think of it like a tiny, coiled hypodermic needle inside the spore. Under the right trigger conditions, it shoots out with considerable force. It literally punctures the host cell membrane and injects the parasite's infectious contents, called the sporoplasm, directly into the host cell's cytoplasm.
Wow, like a biological injection system.
Exactly. It's an incredibly efficient and precise way to invade a host cell. Once inside, the sporoplasm begins to multiply, eventually forming many new spores. These new spores eventually fill and rupture the host cell, releasing them to infect more cells or to be shed from the host to infect new individuals. A well-known example is the genus Nosema. Various Nosema species parasitize many different insects, including silkworms, where they cause a disease called pudbrain, which nearly wiped out the European silk industry in the 19th century. And other Nosema species infect honeybees, contributing significantly to colony stress and sometimes colony collapse disorder. So while you'll likely never see them with the naked eye, their impact can be quite significant. They represent another fascinating, if somewhat sinister, corner of the fungal kingdom, highly adapted to a parasitic lifestyle at a microscopic level.
Wow, what a journey through just these three phyla of Eumycotan fungi. We've covered such amazing diversity and success. From the indispensable recycling power and frankly bizarre dispersal mechanisms of the Zygomycota, to the absolutely vital plant partnerships of the Glomeromycota, and even the tiny, super specialized parasitic Microsporidia. These organisms are truly masters of adaptation and so crucial for life on Earth.
This deep dive really does underscore how fundamental fungi are, doesn't it? And it raises that important question. If we've only really scratched the surface of fungal diversity, with potentially well over 1 million species still awaiting discovery, what other incredible roles or maybe challenges might these organisms present? Their adaptability, their enzymatic prowess, their intimate involvement in ecosystems and, our daily lives, it's truly astounding. They continuously challenge our understanding of life itself and just how interconnected everything really is.
And it all starts with those tiny, ubiquitous spores and those incredible exploring hyphae constantly reshaping the world around us, often unseen. This deep dive into just a part of the Eumycotan fungi truly highlights how much we still have to learn from this hidden world. With that, we, your guides from the Deep Dive team, want to extend a warm thank you for joining us on this exploration. Keep that curiosity alive, and we'll catch you on the next Deep Dive.